selectable markers for animal cells. So previously we have discussed that variety of selectable markers that may be based on antibiotic resistance, they can be used for the selection of transformants. अगर आप बैक्टीरियल ट्रांसफॉर्मेशन को जहन में रखें, हमने बहुत पहले डिस्कस किया था कि बैक्टीरियल सिलेक्शन के लिए हम एंटीबायोटिक्स के मार्कर बड़े कॉमनली यूज करते हैं. तो सिलेक्टेबल मार्कर्स जो हैं ये हेल्प आउट करते हैं कि क्या हमारा जो सेल है इसने फॉरेन डीएनए को अपटेक कर लिया है या नहीं. तो इस पर्पस के लिए हम एंटीबायोटिक रेजिस्टेंस जीन जो है वो भी उसमें रखते हैं हमारे रिकॉम्बिनेंट जो प्लाज्मिड है, ताकि वो हमें इंडिकेट करे कि ये ट्रेट बैक्टीरिया के अंदर आई है कि नहीं. वो इंडिकेट करेगी कि हमारे जो सेल्स हैं वो ट्रांसफॉर्म हो चुके हैं कि नहीं. Similarly, we have also focused on plant cells that after transformation they will be regenerated into callus, or as a result of organogenesis they can be developed into whole plantlet, and their selection it is also based mostly on antibiotic resistance characters. If we consider the animals, then you can say that what type of selectable markers can be used. Again, here in case of animals we have variety of selectable markers. For example, if we focus, then we can say that some endogenous selectable markers are already present in the animal genome. For example, some markers in case of the synthesis of some enzymes, they can be used, and here one of the enzyme, it may be thymidine kinase or TK enzyme. And here if the wild type phenotype is there or wild type cells are there, then naturally wild type cells, they have the ability to produce this thymidine kinase or TK enzyme. And if we are going to use some TK enzyme deficient cell, it means that the gene encoding this thymidine kinase, it is mutated, it is no longer have the ability to synthesize this vital enzyme. And if we will transfer our gene of interest along with the gene that encodes thymidine kinase or TK enzyme, then the transformants, they will also have the ability to produce this TK enzyme due to the presence of this gene. So wild type phenotype will be restored, and such a type of transformants will indicate that foreign gene, it is also present along with the gene that encodes a TK enzyme. So this is one of the example. And earlier we also focus on Hprt enzyme. Hprt, if you remember, it stands for hypoxanthine guanine phosphoribosyl transferase. And this enzyme can also be used as a selectable marker. And the most important point is that these two enzymes, they are very important for the salvage pathway. If you have the idea that DNA it can be synthesized as a result of de novo biosynthesis by using some sugars or amino acids as a precursors, and sometime by using the salvage pathway, the already existing nucleotides, they are recycled. So for the salvage pathway where the nucleotides they are recycled, here these two enzymes like thymidine kinase or TK enzyme or Hprt enzymes, they are most important for the recycling of the nucleotides. So such a type of cells that have been transformed by using the genes of these enzymes, they are grown on a HAT medium that contain an antibiotic aminopterin. so that the de novo biosynthesis of nucleotide is inhibited, and cell will only grow if these genes for TK and Hprt enzymes they are present. So this character, which is endogenous, it can be used as a selectable marker. And finally, like the bacteria and animal antibiotic resistance that can encode some phenotypic traits or dominant selectable markers, they can also be used. For example, here we can say that some antibiotics like neomycin or kanamycin that can inhibit protein synthesis in animal cells, they can be used as a selectable markers.